The number of people diagnosed with Alzheimer's is increasing and places a heavy social and financial burden on families and society. Dr. Lunte and Dr. Culbertson are working to better understand the underlying causes of Alzheimer's so that treatments can be found.
With the devices that we've developed in collaboration with Sue, essentially we can take a single cell, rapidly lyse it, and inject that cell lysate into a channel the size of a human hair. We apply an electrical voltage across that channel, a couple thousand volts, and separate out those components in a few seconds. And we can quantitate then the amount of these reactive oxygen and nitrogen species in the cells. And then we can perturb the cell and see how those concentrations of reactive oxygen and nitrogen species change over time.
Electric field strength that will allow us to separate the analyte into the channel in a very fast speed.
Dr. Lunte, Dr. Culbertson, and their students are looking at the chemistry of cells and their connection to inflammation in the brain. They are trying to develop a deeper understanding of the effects of reactive nitrogen and oxygen molecules on cell physiology and how these molecules cause inflammation. By using electrochemical and microfluidic technology, they can pull apart a single cell and observe its characteristics.
If we can understand that, then we have a better handle on how to potentially develop a variety of potential pharmaceutical agents to treat or prevent Alzheimer's disease, ALS, Parkinson's disease.
So in a device we have sample reservoir and a waste reservoirs. So in between those reservoirs we are running single cells. Those single cells are labeled with fluorescent dyes. Those fluorescent dyes can specifically capture certain molecules that we are looking for. For example, nitric oxide, superoxide, and that kind of molecules. You can do electrochemical detection. So collaborating with
other researchers in terms of what molecules, what proteins that they should target in a cell to prevent these diseases. So we're looking for pharmaceutical targets in these cells.
While I was at the University of Kansas, I took a tour of Dr. Lunte's research labs. My main focus was to see what kinds of experiments Dr. Sue Lunte and her graduate students were performing in their labs. I also wanted to see how both K-State and the University of Kansas benefited from the collaboration of this research. I talked with Dr. Lunte and Dr. Culbertson together to get a little better history of the project. Dr. Lunte and Dr. Culbertson's research could have a potentially widespread impact in the medical and pharmaceutical field. Their work in identifying free radical molecules and possible inhibitors of free radicals could give doctors and pharmaceutical companies an idea of how to target brain cells in order to prevent the release of nitrogen and oxygen species and decrease neuroinflammation. Dr. Lunte and Dr. Culbertson's work using electrochemistry and microfluidics to study single cells could help families and their loved ones who are suffering from the ravages of Alzheimer's, ALS, and Parkinson's.